We'll start at 12:05 or 3:05, depending on where you're signing in from. So 2 more minutes to get a couple more folks in. Okay, we're going to go ahead and get started. Well, welcome everyone. It's a pleasure to have everyone join in today. My name is Sangeeta Iyer. I'm the scientific advisor for the ACD. I'm joined today by, on the ACD side, by Erin. She's a parent board member of the Association for Creatine Deficiencies. And we're gathering here today to hear from Ultragenyx as well as our Vigilant PI, Dr. Judith Miller. And just to provide a little bit of context for everyone who's gathered here, if you have stayed up to date with the news arising from the ACD and the CCDS community, you will be aware that a decision was made earlier this year to close the natural history study ahead of schedule. This information was released sometime in September. And this step was really taken as part of the natural progression of the natural history study that was underway, titled Vigilant, which was sponsored by Ultragenyx. And Ultragenyx had circulated a letter as well, providing context around this decision. Today, the team that was responsible for the Vigilant study on the Ultragenyx side, as well as the NIH side, will be providing additional information relating to the findings from the Vigilant natural history study. Following this, we will have a Q&A session with the pre-submitted questions. So when you registered, you had an opportunity to submit questions relating to the natural history study and the decision as well. So these will be addressed post an update from the team. And if attendees have additional questions during the course of the presentation, please feel free to put them into the Q&A box. We will be compiling this for our panelists. We'll have these addressed offline and provide a written response associated with them after today's event. So without further ado, I'll hand it off to our panelists after a short introduction. So the update today will be given by Dr. Melanie Brandenburg. For those of you who may not be familiar with Melanie, she is currently a senior medical director in the global clinical development team at Ultragenyx. She works on the development of therapeutic agents for rare diseases. She's built a strong clinical career as a specialist in Parkinson's disease movement disorders, and has also served as the medical director for 3 National Parkinson's Foundation Centers for Excellence at the University of Illinois, at Alexion Neurosciences Institute, and the Parkinson's Institute in Sunnyvale, California. She will be joined by Dr. Judy Miller, who is the coordinating principal investigator for the Vigilant observational study of creatine transporter deficiency. Dr. Miller is a clinical psychologist with 25 years of experience in developmental disorders. She has a joint appointment as the assistant professor in both the psychiatry and the pediatric departments at the Children's Hospital of Philadelphia, or CHOP, which is affiliated with the Perelman School of Medicine at University of Pennsylvania. She's also the Clinical Training Director at the Center of Autism Research and Associate Director for Leadership and Education in Neurodevelopment Disorders Program at CHOP. So that, Melanie and Judy, please feel free to take it away.
Thank you so much, Sangeeta, for that very nice introduction. So I think Dr. Miller and I will sort of tag team this, and hopefully that won't be too awkward and we'll get through this relatively smoothly. We're going to give you an update on the Vigilance study. And first and foremost, I want to emphasize that this was an extremely successful study. We've gotten lots of data. We've learned so much that will help not only our team, but any other investigator working in this field. So everyone who participated, we're exceedingly grateful, and you should feel very proud that we have come through this with such a great amount of data due to your hard work. So thank you. So we were just introduced, that's us. So today's focus is to discuss recent updates to the Vigilance study, to share some preliminary results and learnings from that study, and address questions that we received in advance from the CTD community. Questions submitted by the CTD families to ACD have directly shaped this presentation. So we're trying to sort of preemptively answer a lot of the questions we received in the way we've structured this presentation. So we are going to talk about the role of natural history studies in therapeutic development, the drug development process. We are going to talk about Vigilance specifically, the history, the goals, and the stakeholder participation, and that includes the patients and families who participated and the expert clinicians who participated, as well as Ultragenyx. And then study closure and preliminary results and learnings from the study. Then we will talk about the implications on the study data for future research and next steps into hopefully developing a treatment for CTD. Then there will be moderated questions and answers. We have some questions that were submitted. We've put together some answers, and hopefully it will help answer some of your questions. Next. We did receive 10 questions from the community, and we will answer all the questions that were submitted. Some of the questions, though, will be answered in the presentation. And then when the ones that weren't specifically addressed in the presentation, we will have a moderated Q&A session following the presentation to make sure that the community has access to clear and understandable information. So here are some of the questions, and I'm not going to answer these questions now because, as you'll see, some of them are answered in the presentation, and then some will be answered at the end of the presentation. So we had a lot of questions about the timelines for what for us is called the UX068 program. Is there success in the drug development? Are there eligibility criteria for future studies? When are the next steps? What is the next step in involving patient participation? When are trials planned? Will the observational data collected be available to other researchers? Is there not value in continuing to collect data to see how CTD manifests over time for longer than just 4 years? Did the study end early because of budget cuts? With such an abrupt end to the Vigilance study and the patient's final visits being a bit rushed, does this impact the integrity of the study results? And how does ending Vigilant early specifically help or improve the UX068 clinical development program? So again, listen for answers to these throughout the presentation, and then we'll have some additional time at the end of the program. Again, I want to thank the Vigilant families. This research would not be possible without you. The Ultragenyx team and the entire Vigilant study team are extremely grateful to the patients and families. You have helped establish data that will help us understand the disease manifestations over time, data that will shape research today and in the future, and the value of that cannot be overstated. Next. I think, Judy, I think this is yours.
Sure.
Am I correct?
Sure. Yep. I can take over from here. Okay.
Great.
All right. So just briefly, that was really great lead up, and I'm really excited to be here. So I'm thank you everyone for coming, and thank you for giving me the opportunity to participate in this project. I don't I don't work for Ultragenyx. I hope that was clear from the introduction. I work at the Children's Hospital of Philadelphia. This is a project that is near and dear to my heart. So it's been a fun collaboration with the families and with Ultragenyx for this. All right, so why do we do natural history studies? We do them to look at how a disease progresses over time when there's no intervention. That's the definition of a natural history, is what happens in just nature with this disease. And from that, you can start to look at, are there any natural variables like demographic or genetic or other variables that impact outcomes in the absence of any intervention? And that's really important to know for understanding the disease and also understanding any treatment. So it's particularly important in rare diseases because it's really important to know what is your baseline level of development in this case, or health in other diseases. And then you can use that to compare a future outcome measure and understand if an intervention that's happening in between is working safely and if it's working effectively. One of the challenges with rare diseases is that it's very hard to conduct a randomized control trial because a rare disease doesn't happen in very many people. So it's hard to fill a study where you need a whole group that is going to just not get intervention and a whole group that's going to get intervention, and that both of those groups are big enough that you can really understand how an intervention is working. It's really hard when there's not very many people with the disease. That's different than when there's a common disease. So when there's a common disease, you have so much information. People from all over the world may have this disease, and there's a lot of people with it. So there's a lot of opportunity for many scientists to study the populations and to contribute information. In a rare disease, that's just very much more limited. So you really have to take a very planful approach about how are we going to understand how this disease progresses in nature, and we have to do it in an organized way. You can't assume that that it will happen organically. Next slide. All right, so just a little history of the Vigilance study. The original sponsor was Lumos Pharma. They originally planned this to be a 2-year observational study. And the idea was they were quite far along in their drug development, and they wanted to invest in a natural history so they could understand how to measure their intervention. So they reached out to me and to other folks to help them conduct this 2-year observational study. We knew it would take more than 2 years to recruit enough participants and get all the sites involved that needed to be, but we started enrolling participants in 2016. Along the way, as Lumos was developing their intervention, they decided that they made the decision to extend the natural history study because they were working on their preclinical studies that included animal work, and they had encountered some obstacles and they were still working through them. At that point, things seemed to still look promising. So they decided to extend the natural history to a 4-year study, which would allow maybe a more seamless progression from the natural history study into a clinical trial. And the sites were already up and running. The families were already mostly recruited. And so it just seemed like it was a good opportunity. But then as they were working through those preclinical and animal studies, they unfortunately encountered some safety issues with their intervention that really couldn't be remedied. They spent I really think they exhausted all possible options to overcome those safety hurdles. But in the end, they just couldn't figure out a way around them. So they had to discontinue their drug development. They offered the Vigilance study to Ultragenyx because Ultragenyx was doing a completely different intervention idea. And this was maybe an opportunity to keep Vigilant going, and also for Ultragenyx to be able to benefit from all the work that had been started with Lumos. So Ultragenyx assumed sponsorship of the study in 2019. Next slide. All right, so the goals for Vigilant originally were to enroll 50 males with CTD for a 4-year study. Again, originally it was 2 years. The purpose was to enhance our understanding of symptoms and the course of people living with CTD, contribute to developing therapeutic interventions because we would understand what the natural history is. We would be able to identify potential study endpoints that were relevant. So as we learned about families and individuals with CTD, we could really learn what are the relevant endpoints and how can we measure them. It was also a great opportunity to develop relationships with the families and the sites and the physicians that you need in order to conduct a future intervention trial. The purpose right from the outset also was to engage with people living with CTD and the organizations who support them to understand the community and all that goes along with raising and loving and caring for folks with CTD, and then to disseminate what's been learned to researchers, clinicians, and families and advocacy groups. Next slide. All right, so here's a little timeline. So as I mentioned, we started enrolling in 2016. So it was probably a year or two before that that we started designing the study. And the first sites were CHOP and NIH. And then you can see it was 2018 when we had established the 10 sites that were involved, and we had enrolled almost all of the 50 participants. The transition to Ultragenyx was in 2019. In 2020, we were ready to do a preliminary sort of interim analysis, and then the pandemic hit. So we still conducted that, certainly took a lot longer than it might have otherwise. But that's when we started doing the interim analysis, looking at what data did we have and what could we learn from it so we could know if we needed to make any final revisions before the study really ended. In 2021, we kept finding that our data results were very consistent. Every interim analysis was very consistent with the last one. Every iteration, I mean, was very consistent with the last one. And we've presented this data at a couple different conferences. And at this point, what's happening is the results are the same, but the sample size is bigger. So very stable results, which you'll see shortly. In 2022, at this point, we have 43 of our participants have 2 years worth of data, which was the original study length design. 34 have 4 years of data or will have by the time they're done. And there were about 6 patients who we could have had more data from, but won't get all their data because the study will end early for them. That does mean that there are some families who weren't going to finish 4 years anyway. They moved, they could no longer travel, or there's a couple of sites had to modify and close. So when you have a study that goes this long, you have some natural attrition anyway. So big picture, we have a lot of participants with 2 years of data, a lot of participants with 4 years of data, and about 6 patients whose study visits were impacted by the early closure. Next slide. All right, so here's just a map of where we drew our participants from. There it is. It's very exciting. All right, next slide. This is the study team. These are the sites that have been involved. A couple of them are inactive at this point, or ended. And their participants had an opportunity to transfer to another site. But as you can see, this is a very big team. It's been really fun to get to know all these investigators and work with this. But this is a big machine for this study. It's a lot to have 10 sites happening. Next slide. All right, this is a slide about information about CTD. So we can come back to it if you have questions, but we know a lot about the genetics of CTD. Just briefly, we know that CTD is an X-linked recessive disorder. The mutation is SLC6A8. It's one of the leading causes of X-linked intellectual disability in males. We're learning more and more about how it might look in females and that phenotype and how it ranges. There are a lot of variants of CTD, and there's definitely more to learn there, but in the end, it does seem to be all on the same gene, so we can identify it pretty well. The clinical presentation is there's neurological challenges, there's seizures. Many people with CTD also have autistic features, a lot of speech and language disorders and delays. And many people with CTD have behavioral problems and challenging behaviors, intellectual disabilities, motor skill delays, some extrapyramidal symptoms. Brain creatine levels can range, but they're usually very low. So they can be undetectable to just about 20% of normal. So very low level of creatine in the brain. And there are some deficits that are not necessarily related to the central nervous system, but muscle hypotonia, hypertrophy, and global developmental delay, and some failure to thrive. A lot of folks with CTD have an early history of feeding difficulties and failure to thrive. Right now, there is not really a standard treatment that's effective. It's probably underdiagnosed. Sometimes that's because there's no treatment, so it's hard to raise the awareness enough that there is a treatment, which then raises the diagnosis. But right now, the care that we offer for families with CTD is more supportive. It's therapy like physical therapy, occupational therapy, speech therapy, behavioral therapy. And there are some treatments for seizures. Generally speaking, most folks with CTD and seizures, the seizures can be managed pretty well, not perfect, but in general, seizures are pretty manageable. Next slide. Okay, so we're going to quickly go through the data that we have, and this unfortunately is not in lay language. These are the slides that we have presented at scientific conferences. Some of those scientific conferences parents also attend, so hopefully this will make sense. You should know that we are actively working on the manuscript for this, and that will come with a lay summary, and we will also talk about some more simplified language that we're going to use to be able to summarize all this. But right now you're getting exactly what the scientists get. So let's do our best together. All right, so our participants, this is the distribution of age. So this slide tells us that most of our CTD Vigilant participants are under the age of 12. So about 78% of them are under the age of 12. And then we have a couple adolescents and a couple adults. Next slide. This slide tells us, we asked everyone, when was your child diagnosed? But also, now that you know it's CTD, if you look back in hindsight, when do you think the very first symptom was? So that's a really important question. And because if we want to be able to identify it earlier, then we need to know maybe when was the first possible symptom? And in our sample, the first possible symptom was at about 6 months of age. Not for everyone, but as you can see, when it says median there in that first box, that means that half of the kids in our sample had their first symptom before 6 months of age. Doesn't mean that people understood what was happening at the time. This is in hindsight. But so at least half of the kids, there was something going on at 6 months of age. The next box over on the right, the age of initial evaluation. So this means like in real time, as your child was developing, when did somebody evaluate your child? And the median there is 15 months. So that means half of the kids had some sort of evaluation at around 15 months of age. Usually it's for something that's not very specific. At 6 months, most of the concerns are kind of not very specific. There's a general delay. At 15 months, you're getting evaluated. There are some missed milestones. It's not anything that just is very, very specific to CTD. It's kind of general. Okay, then the third box, bottom left-hand side, time from initial evaluation to the CTD diagnosis. So from that 15-month evaluation to a diagnosis of CTD, how long did that take? That unfortunately took about 2 years for half of the kids. So the median there is 24 months. So there's a lot of things that have to happen from that first evaluation to when you get the genetic testing that's indicated it was CTD. So there's a lot of room for improvement there. Sorry. And then finally, the last box, the age of CTD diagnosis. How old was the child when the CTD diagnosis was made? And the median there is 48 months. So that means that half of the kids had their diagnosis by the age of 4, but that means half of the kids got their diagnosis after the age of 4. Okay, next slide. Okay, now I'm going to give you a quick and very high-level view of some of the characteristics of our sample with CTD. So this shows seizure history. Now, you couldn't get into the study if you had active seizures that were not controlled. So this is not representative of everyone with CTD, but of our participants, 68% of them had a history of seizures. 30% had not had a seizure. And there's a table that tells you the average age of the first seizure is about 4 years, almost 5 years. So it's, again, it's not totally descriptive of the whole population because you couldn't have active seizures and be in the study. But you can see there's definitely a seizure history in a lot of our kids. Next slide. This is features of autism. So this pie graph is showing us that about 1/3 of the kids in our sample had already been diagnosed with an autism spectrum disorder. So about 2/3 had not. You'll see later on, there's a lot of autism characteristics in a lot of kids with CTD, but not everyone. So this is about 1/3 had already had a diagnosis. Next slide. All right. We did a lot of developmental testing through Vigilant because that was what we were trying to figure out. How do these kids develop over time? So every 6 months, we did testing on the participants, and these are the results. And we'll go through them. Next slide. There'll be a lot of data on some of the slides, but I hopefully will be able to highlight the most important parts. Okay. So what did we measure? So Lumos had done some pilot work, and we had surveyed parents about what their priorities were. And the priority was very clearly around functional skills, independence, communication, that kind of thing. So we had a variety of testing, standardized tests in order to look at that. So from a developmental standpoint, we used 2 measures that are kind of getting at development and intellectual functioning. So the Mullen is like an IQ test for very young children. It's widely used in a lot of research. The Wechsler abbreviated scales of intelligence is a brief IQ test that is used in schools and all over. It's one of the most widely used ones around the world. So we used both of those to try to understand verbal and nonverbal concept formation in our kids with CTD. Then from an adaptive skills testing, adaptive skills are independent skills. Can you get dressed by yourself? Are you toilet trained? Can you cross the street safely? Can you make a phone call? That kind of stuff. So we used the Vineland adaptive behavior scales as an interview. So we interviewed all the parents every 6 months. What's your child able to do right now? The Vineland measures communication, socialization, and daily living skills, like I mentioned. And then the Vineland has a feature in it where you can compare someone to themselves earlier. So many, this is unfortunately not available in every standardized test. You have to build it in from the beginning when you design the test. Some educational tests have this that are used in school settings, and it's increasingly getting built into tests, but like the Wechsler doesn't have it. The Mullen doesn't have it, but the Vineland has it, where I can do your interview now. And if I do your interview every 6 months, I can compare every 6 months to your original, or I can compare every 6 months to your last 6 months. And that's really great for intellectual disabilities because you may make a lot of progress, but maybe you're not going to make you're still not going to make as much progress as a child without an intellectual disability, but you are going to make some progress. So we want to be able to measure that. And it's also great for rare diseases because we don't want to compare you to every child under the sun. We'd love to be able to compare you to the people who are most relevant to your situation. And who's more relevant than yourself? Nobody. All right. So that's why we're excited about the Vineland. And then third, we measured behavior. We measured autism spectrum characteristics and challenging behaviors, because that was a concern for many families. All right, next slide. Okay, the Mullen and Wassey, because they don't have this, this feature that I was talking about, what we're left with is the regular scores that you get when you give these tests to any child. So the average of an IQ test across the nation, they're statistically designed so that the average IQ is 100. So if you have an IQ of 100, that means you're perfectly average, which is great. You're right in the middle. Most people have an IQ between 85 and 115, and anywhere in there is considered perfectly average. So what this shows us, we know that kids with CTD have intellectual disabilities. We didn't fully know, like, is that something that changes over time? Does it get better or worse, or is it pretty stable? What we found in our sample is that it's pretty stable. We're limited in that we can only look at these standardized scores, which again are kind of statistically designed to sort of not be super movable, but our kids were pretty consistent over time. What you see from this, if you can notice, is just the scores are just hovering around 30. That's kind of where they're staying, which means that as a group, the kids with CTD are pretty intellectually disabled, and it stays that way over time. Next slide. Okay. Oh, and just to remind people, okay, so maybe go back one slide. So an average score is 100. When you're getting below 70, you're in the range of what we call an intellectual disability. And when you're getting lower than that, then you move from mild to moderate to severe to profound. So a score of 30 is kind of in the severely intellectually disabled range. Those bars you see where there's the black line with the little edges, that's the range. So there are some people who were getting IQ scores closer to 50 and some who were getting closer to 20. And so you can see there is some range in there, but in general, this population is between the moderate to severely intellectually disabled. Okay, next slide. All right, now let's talk about their Vineland scores. So Vineland is, again, your adaptive skills. What's your independence level over time? So this is the one that you can see some changes or you can compare it to your previous scores, but you don't have to. So we're going to start with the regular standardized scores. And as you can see, they, like the IQ scores, they're very stable. So over time, we're seeing very consistent intellect or independence skills. They're hovering between that 35 to 65 range on average. Each of those lines is a different type of skill. So the bright blue line is communication. The mustard yellow line is daily living skills. The darker blue line is socialization. The light gray line is motor skills. And the purple line that's kind of right smack in the middle, that is the overall score. So when you take all those things together, the average is about 50 and it's staying pretty consistent across time. Next slide. All right, so like I mentioned, we want to see if we can find something that we can measure as a valid endpoint. And we were excited about the Vineland because it is an area that's important to families. Right now, the scores look like they're showing stability over time. So it's a it's an area that you can measure reliably. You're not going to get wildly different scores from one time to the next just because the day is different. You know, it's not that some days your child is able to do lots of things for themselves, and then other days they just really can't do anything for themselves. Their skills are pretty consistent over time. I lost the slides. There they are. All right. In the Vineland, there are some, these are the opportunities. These are the candidate domains, or the like things that the Vineland measures that we could focus on. So you'll see there's big picture ones. There's communication, daily living skills, socialization, and motor. Within each of those, you can also get a little more fine-grained. So in communication, you can measure receptive, expressive, and written language. Daily living, you can look at personal self-help, domestic, like what can you do around the house, and what can you do in the community. Socialization, you can look at how well are you able to interact with other people, what are your play and leisure skills, and how well are you able to cope with things that don't go quite right. And then in motor, you can look at fine and gross motor skills. Next slide. So we looked at all of those, however many there are, 13, 3, 6, 9, 11. There's 11 of them. We looked at all 11, and we looked at this score I'm talking about that you can compare to previous ones. It's called a growth scale value, or a GSV. And this plot is really just to show you this is every single participant and their scores over time. And what you hopefully can see, though it's subtle, is that if you look at everybody, most people are improving at least a little bit over time. So nothing dramatic, but you can generally see kind of an upward trend. That's kind of what we are going to use to try to see how can we learn from that, and how can we use that in a future intervention study. So we can see some growth. It's subtle, but it's enough that we could measure it. All right, next slide. So actually go back one. I think, what was that domain? That was receptive language. So just one of those 11 things. We have to look at each one. You can't clump them all together. Unfortunately, you have to do each one separately. So receptive language is making a little bit of gain over time. Next slide. Same idea with personal independent skills. So things that the child is learning how to do for themselves. It's slowly getting better over time. And this fits with what parents tell us, like from one visit to the next, they recognize things the child is doing that they weren't doing before. But overall, the kids still need a lot of support. So you can see the slight gradual improvement in personal skills. Okay, next slide. Same idea with expressive language. You're seeing, there's some fluctuation there, but you're seeing general sort of upward trend. Next. And then coping. Coping's a little bit all over the place, but coping is an important skill. And even though this looks really messy when we analyze it all together, it does look like there is some growth over time. All right, and those four, there's 11 possibilities. Those four are probably the four that kind of seem like the best candidates, because they're very meaningful and they're developmentally appropriate. One of the things I mentioned was written communication. That's not really appropriate. That's not the most important thing in a toddler with CTD. Community, self-help skills, that's not really important either, but expressive, receptive, personal skills, and coping are all very, very relevant and very important. Okay, next slide. Okay, so that's the end of the Vineland. So that's the end of the adaptive skills. The next chapter is on behavior. So we have a couple measures of this. The first one is the aberrant behavior checklist. This is a measure, it's a parent report scale. It was designed for kids with intellectual disability and was designed as a way to look at some of the more challenging behaviors that people with ID sometimes have, and that create problems for participating in things. So from what you can tell here, if you notice that there's a blue line and a mustard line that are kind of higher than the rest. And if you look down at the bottom, the blue line is stands for IRR, which doesn't mean anything unless you know that it's irritability. So that is the irritability subscale of the ABC. And the mustard line is hyperactivity. So what this is telling us is that irritability and hyperactivity are the most challenging behaviors for our participants. The other three, which were low, and they weren't really a problem for everybody, is stereotype behaviors, inappropriate speech, and social withdrawal. So the other thing, so if you're having challenging behaviors, you're most likely to have problems with irritability and hyperactivity. If you notice these bars with the edges again, so those are sometimes called error bars or range bars. If you notice how big they are, what they're showing you is that there's a lot of variability in this group. So not everyone with CTD is irritable and not everyone is hyperactive. So what we learned from this measure is that if you have challenging behaviors, you probably are irritable or hyperactive, but not everyone with CTD is. So this would not be a good measure to be your first measure in a clinical trial, because we have plenty of kids who came in and scored low across the board on all of these. So it's helpful to know, and it's helpful in your understanding of the child, but it was really important that we looked at this over time and really came to see this is not where to put your intervention outcome measurement, because not everyone starts out with challenging behavior. Okay, next slide. Another thing we looked at was autism spectrum characteristics. And this shows you that we kind of found three groups. So one group scored 12 or higher, which on this measure, this is a parent report measure, that means you have a pretty high likelihood of having autism. It's hard in an IED population to know for sure without a lot more information, but you have a lot of autism characteristics. Then we found another group that had scores between 12 and 15, which means that, oh wait, no, I was backwards. Sorry, I was going backwards. The blue box is the kids below 12, which means they really are not showing autism characteristics. So yeah, they really just don't seem to have those autism characteristics. The green box in the middle is the group that might. Their score is between 12 and 15, and they are showing some characteristics, but maybe they're mild, maybe they just haven't fully manifested yet. And then the orange box at the end, the group with 7, they have a score over 15, and that is a stronger indication that they have an autism spectrum disorder. If you remember, a third of our kids had already been diagnosed with autism, so we expected at least a third of the kids to come up high on this measure, and they did. And so what this tells us is that autism is not in everyone, but it is in a lot of kids with CTD. One of the most important things that this and the ABC, the irritability and hyperactivity measure, tells us is that if we want to study an intervention, we're going to need to know whether the child also has irritability, hyperactivity, or autism, because that may impact how well that intervention works. If we're giving a kid a medicine and then we're putting them in an educational opportunity or an occupational therapy opportunity, if they also have autism, for example, then that also may make it hard for them to learn. And it may be that the intervention was addressing the CTD part, but the autism part was still present and impacting what the child was able to learn down the road, right? So we need to understand it so we can make sure that we don't miss an opportunity to find an effective intervention that works, that is addressing the CTD, even if there's other things happening at the same time. So it just helps us understand the whole picture, understand everything that's going on, so we don't undervalue a potential intervention. Next slide. Okay, We also wanted to understand some of the medical and physical things that happen over time. We had families for 2 to 4 years, so we asked them about other things that were happening, and we tracked whether there were any medical events that happened. We weren't doing any intervention, so there was no concern that the study was causing any problems. Some of the kids did have challenges when they underwent some of the scanning procedures, because they had to be sedated and they had to get an IV. So things that happen when you do that happen for some of our kids. But in general, we were kind of looking at just over the course of time, what happens. So next slide. All right, so we asked everybody about GI symptoms, and as you can see from this, so if you go down to the bottom, it will tell you green is happening currently, blue has happened in the past but isn't happening now, and yellow is it's never been a problem. So this is ordered by the top row is the lowest frequency problem, and the bottom is the most frequent problem. So in our kids, the most frequent problem is constipation, with a lot of kids that's still struggling with it. Next comes choking and gagging, vomiting, GERD, gas, that kind of thing. So we know from a lot of work that feeding issues are hard, and it also looks like constipation is a pretty big challenge for a lot of the kids. Next slide. All right. We have like 3 slides that are going to come up, and they're just these very long lists of all the different medical events that we looked at. This is mostly to help you see that there weren't that many. So if you look over in the right-hand side, we've got, the 50 kids, and the number of participants with a serious medical event was 8 over the course of the 8 of the 50. So, and the serious medical event is something like a seizure, or like a significant illness, or sometimes it can be, well, you can see the list here: vomiting, GI problems, agitation, aggression, that kind of thing. Okay, next slide. So you can see there's not that many people who have them. And so that's just really helpful to know. This slide is the people with any medical event. So the first slide was kind of like serious medical events. This one is kind of just anything. So how many kids are getting regular just GI problems, regular pneumonia, regular upper respiratory things? And you can see there, you get more of them. So 34 of the 50 had some sort of medical event over the course of 2 to 4 years. So that's pretty, it's still not as significant, like this is not happening every day. Next slide. Okay, and then here's a summary of like the most frequent medical events happening over the course of the study. So it's mostly respiratory problems and allergies and dental things, and a lot of things that you would see in kids. Next slide. Okay, so what did we learn? We started doing interim analyses right at the beginning of the pandemic. What have we learned? So we know that our patients with CTD are showing pretty significant and pretty persistent impairment across all of the constructs that we've studied in vigilance. So their cognition, their behavior, their communication, their motor functioning, and their daily living skills. Pretty even and pretty consistent. We learned that the assessments are pretty consistent. So that's good. It is challenging to test a child with a significant intellectual disability. They don't always understand the instructions. You want to make sure you're giving them the best chance to show you what they know how to do. But they can get frustrated because they're being asked to do really hard things, and it's not clear, and you have to follow you can only say the directions a certain way, because that's how the test was designed. So it's a challenging opportunity for the child. And what we learned is that even given all that, the kids really do try to participate, and even with the challenges they have participating, we're getting pretty consistent results. So that's nice. It's pretty reliable. The implications for that for a drug development are that we do have several assessments that hopefully will be able to help us understand whether a treatment is effective or not, which is nice. We have learned that which is really important, that these measures with a growth scale value are really important, and for measuring not only just natural history observations, but I think would be really important in an intervention trial. It's a kind of a newer concept for the FDA to recognize, but I think there's a growing awareness. We're not the only people to find this, and we're not the only disease where this would be important. So I think, but it is relatively new for a regulatory agency like the FDA. We also learned that the clinical presentation is really important. We need to know about it because that may also impact an intervention, and we don't want to miss a helpful intervention because we were not understanding all the things that were contributing to a child's strengths and weaknesses. All right, and then we also sort of got a sense of what medical events you can kind of expect from this population. Granted, this is a population that doesn't have a lot of active seizures when they started, so it may not represent everyone with CTD, but we found kids who over the course of 2 years didn't have a lot of medical events. Next slide. All right, we also learned a lot of things that didn't work. So in the beginning, we had measures, we have learned that it didn't really work very well to ask kids to wear anything. A lot of our kids won't wear a hat, but we tried to get them to wear an EEG cap, and that got better each visit, but it never really got easy. So we also tried using trackers, like where we would have a tracker on a watch band or put it on their wrist or their ankle. And, these kids are really good at taking things off. And so that is probably not the best way to measure things. We also learned pretty clearly that following really precise directions, verbal or nonverbal, is really challenging for these kids. If they know what you want them to do, they try, they do it. And you can see it, like you can tell, like when they can just tell what they're supposed to do, if they know how to do it, they will definitely try to do it. But if they don't know what you're asking, or if they don't know how to do it, they get frustrated, and it's very hard to guide them through it. It's hard for them to just imitate you because you say, try this, and they do it, or do this, and they just really can't follow those kind of directions very easily. So it's really important to kind of figure out what you're asking, and if the task is one that the kids can demonstrate their best abilities for you. We also learned, we tried several rating scales that were not designed for an IDD population, and we have now evidence that they really don't work for this group. And that was really important because the FDA is really familiar with a lot of parent rating scales that were not designed for ID. And if you go to the FDA and you say, like, hey, I think we have this intervention that could be effective, they are familiar with measures like the ADHD rating scale or like the parent stress index. And they would understandably say, well, how did these kids perform on that? And does the intervention impact those kinds of things? We gave those measures, and we had so many families who said, this just doesn't really fit. I don't really know how to answer this for my child because this clearly was not written for a child with an intellectual disability. And we have lots of data now on that. And we can say clearly, this just really isn't the right kind of measure. And that's super helpful. So we think that all of these things together, the things that worked and the things that didn't work, are really going to help us design a good study in the future. And it's really going to save a lot of time and effort in creating a good interventional study and measuring the right kinds of things. Next slide. All right, so to sum up, the most important implications for future research, we really liked using measures with a growth scale value. The Vineland has that, and it also touches on adaptive skills, which is really important for this population. I think I've gone through all of these points. Independent skills are important. The results were reliable over time, so we think they're accurate, and we could detect some growth over time. And the idea is that if you see an intervention that's effective, that growth will be amplified by the intervention. But here's where we need to have some real significant cautions and caveats. It is also possible that if your scores are that stable over time, that may be very difficult to overcome. So this is true for all neurodevelopmental disorders, not just CTD. But if it's, let's just take an example. If it's been very hard for you to learn how to be fully toilet independent, it may be that that is something that is going to remain hard for you forever. It may be that there's a lot of things contributing to why that's hard, that an intervention just might not be enough to overcome. So we think, so it's possible that the degree of improvement that an intervention study would need to have might need to be really, really significant in order to find that measurable difference. All right. So this is kind of like, there's two sides of this. Stability in scores means that it's not so random. It's not random. So if there's a change, we think that change is real. But stability in scores also means that it might be hard to find change. If the disorder is really, what's the word, unmovable, then it just may be hard to find change, even with a great intervention. Next slide. Okay, so our next steps are, we're working on the manuscript to put in all the analyses and the results I just went over. And we think that's going to be a good resource for anybody doing research on CTD. We will make a plain language version and a plain language abstract that will come out. That will also turn into what's called a clinical study report, which is also translated into plain language for families. And I would imagine that will be posted or distributed through the ACD. Like it says here, what a clinical study report does is describe the methods, describe the result, give you a brief discussion of the key findings. And it's part of what you do when you are creating a new treatment and you send that to regulators. And then we'll start putting that together once we have the last participant done, all the analyses done. We have figured out everything we can learn from this. Well, the first big chunk of what we can learn from this, and finish writing it up. Next slide. Okay, I think this, I can't remember where we transitioned, Melanie, but here's an example of some plain language summaries for clinical study reports on different disorders. So just to give you a visual of what it might look like.
Yeah.
All right.
Thanks, Judy.
Yeah.
I think the next one is more Ultragenyx internal things. Let's see. Yeah, this is the updates. So again, this is an attempt to respond to some of your questions. And those of you who participated in the town hall that we held last December will recall that we sort of lay out what's involved in the drug development process and what is going on with our drug development for hopefully an interventional treatment for to be studied in this patient population. So we sort of updated the slide a little bit. I know none of this goes as fast as parents would like it to go, but we've done a lot of work with preclinical proof of concept. We are working on making the absolute best formulation that we can based on the patient population and disease characteristics. And as many of you know, we've been asking questions that pertain to how patients take oral medications and what the tolerance is for certain kinds of foods and so forth. So we are in the process of developing all of these things. Unfortunately, it all takes time, and we have to follow the science, and it has to be done in an exacting way, because ultimately we are hoping to bring this to clinical trials in patients, and we want to make sure it's a safe drug, as safe as it possibly can be, and delivers creatine effectively to the brain in an efficient manner. So all of this takes time, and I know it's frustrating because we've heard from many, many people that they would enroll their child in a clinical trial tomorrow if we had one, but we're not there yet. So this is our attempt to sort of give you an update on where we are. We've sort of moved, since we talked to you, moving more out of the green zone and into the blue zone. So all of this is very time consuming, and we've got a huge team working on it, really great people who are really devoted to bringing this drug to a clinical trial. So we will keep informing you as things move along. And I think there's one more slide kind of with a little more detail on this. We have key research questions driving ongoing preclinical activities, such as what is the best way to deliver enough prodrug to the brain? How does the drug get absorbed, distributed, metabolized, and or excreted? What oral formulation is the safest for patients and best tolerated? And what are the potential dosing ranges? And you can see on the right, we've listed some of the studies that are going on to establish all of those things. So it's the necessary steps of preclinical development, and this is what takes time. And I know it's frustrating for you because you can't see all the work that's going on behind the curtain, but I will assure you that the work is being done by our excellent and devoted team, and we will get there when it's, you know, if the science allows us, we'll get there when it's a safe and effective drug to bring to the clinic. And I think, is that the last slide here for this part? Yes, so now we're going to move to the question and answer part. So I'm going to project, I think that was a separate deck.
Kristen, I don't know if you have the questions, otherwise I'm happy to pull up the document with them.
Yeah, Sangeeta, those questions are for you to ask Melanie.
Perfect.
There is no slide to project.
Oh, okay, okay.
Okay, sounds great. In that case, let me see. Melanie, are you comfortable with me just reading out the questions, or would you like to see them displayed? I'm okay with either.
I think it's whatever works best for you, but I'm also pulling up my answers to make sure that I have them in front of me.
Okay.
Hang on 1 second.
Okay, well, maybe I'll just get started. And I
Okay.
think you're
Go
right
ahead.
that some of these were addressed in your deck, but for the purposes of completion, just because the wording was different, I'm going to go ahead and read out the question. And that'll give us another chance to contextualize it to the component in the presentation that I addressed that. Okay, so I'm trying to organize this in the same way as we did the presentation. So I'll direct questions relating to the natural history study first, and then we'll move into the component of the drug development program. So to start off, did the study end early because of budget cuts to the CTD program at Ultragenyx?
No, it did not. This decision is aligned with our goals as we look to the future of our CTD research program. We decided to end the study early after assessing all of the data collected as of June 2022. We concluded that we had the data needed from the study to apply to our clinical development plan. A clinical development plan is the document we use to sort of outline our research strategy for an investigational product, and includes decisions related to the potential design of a future clinical trial. So this is an important document for us. It changes over time with data, with new information, and we made the decision that we had enough information from the Vigilance study to inform that process. So as Judy very nicely pointed out, the Vigilance data is helping us to identify both the constructs to measure and the tests that are the most feasible to administer in a future clinical trial. So we will combine this with other sources of data collection, including surveys and interviews, and some of you have probably participated in some of those activities. So the Vigilance data will, along with those other pieces of information, will inform our endpoint development and strategy. And as I think Judy mentioned, we will use the Vigilance data to support future regulatory interactions. So when we go to the FDA and other agencies to discuss our clinical trial designs and our plans for research, we will use that data as supporting data to those interactions.
Thanks, Melanie. Next one on the same topic. Is there not value in continuing to collect the data to see how CTD manifests over time in age longer than just 4 years?
Well, that is a really good question. And I think if we all had unlimited time and resources, we would continue to collect data on patients in all of our programs, natural history data for forever. But as Judy pointed out, when the LUMA study initially began in 2016, it was supposed to be a 2-year study. Eventually, it was expanded to 4 years. And the study's really been going on for quite a long time. So as the study ends now, we have 48 months of data on the majority of participants, which is really a wealth of information that, as I mentioned, will inform all of those other activities. So when we reviewed the data, as you saw, some of the patients had some mild development in some areas. There was sort of a plateau effect for most patients, meaning that the data was very stable over time. So we have this data. We understand the stability of this data across a variety of age ranges, and we feel like we have a good sense of how patients perform over time. So I think, again, this will be a really, really important source of information. And while, yes, we would love, all data is good data, but at a certain point you have to say, we think we have enough data and resources are, now we're sort of moving on to the next stage here.
Thanks, Melanie. Another question relating to the trial, and I think this is more a question of how the different sites maybe received information and cascaded it down. But the question is, with such an abrupt end to the vigilance study and the patient's final visits being rushed through within several weeks, does it impact the integrity of the study results?
Yes, and I understand that did seem kind of abrupt, especially for some of the sites. But we don't think that this will have any effect on the integrity of the study results, because the majority of patients had either already completed the study or were scheduled to end the study in September 2022, which is sort of why we chose that as the cutoff date, because we said, gee, look, at the end of September 2022, we will really have a lot of data on the majority of patients. So in June 2022, we had 26 out of 50 patients who had completed month 48 visits and completed the study. There were an additional 13 patients that were already scheduled to complete the study visits as of September. So really, the majority of patients did complete the first full 4 years, and we do understand it did seem abrupt, but partly because of all the things that had to happen in terms of internal and external communication. It had to be done in a certain way, and I know that was a little unsettling for some people.
Thanks, Melanie. So how does ending Vigilant early specifically help or improve the UO668 clinical development program progress?
Well, I think it will help in 2 major ways. And one is that the early completion of the study will allow us to produce the clinical study report, the CSR, ahead of schedule. And this will make this data available for our clinical development plan versus waiting until the summer of 2024 when we were originally planning to complete all of the assessments in the study. And so the CSR is often completed as a part of a submission process when we are going to the regulators to say, okay, here we have this data, here's our plan. Are you in sync with our ideas about how to conduct the study? And so it'll be really helpful to have that CSR completed ahead of schedule. In addition to that, we will use this data as part of our conceptual model of CTD. And I think those of you who were at the ACD conference in June heard discussions about the importance of a conceptual model when you're studying any disorder, really, so that you understand what is involved in the disease, what symptoms are seen, what's the impact on the family, what things are most important to treat, et cetera. And so having this data completed early will help us, along with some of those other information sources that I mentioned, such as the interviews and surveys, will allow us to build that conceptual model, which then helps determine the endpoints that would be used in a potential future interventional study. So the other thing that will be nice about having this, both the CSR and the manuscript, is that this will inform other researchers as well. So we obviously are going to share this data, and then somebody who's working, say, on another idea or another study will be able to use this data as well.
That's great. So to contextualize, because the next question was, will the observational data collected be available to other researchers? So I think you've already addressed this, but let me summarize.
Yeah.
The interim study results will come out as a publication, and the complete study results will be compiled in the CSR. Is that right?
Yes, yes.
Okay.
That's right.
Thank you. Okay. Next question. When will you share updated timelines on advancing UX068 program to the next stage? Is there success in the drug development?
Well, as we discussed in December, we are currently in the preclinical phase of drug development, which includes conducting animal studies, refining the drug manufacturing process, and doing all the things we need to do as part of regulatory requirements before we can even contemplate any kind of interventional clinical study. So this is something that unfortunately takes time, and we have to follow the science as we discussed in December. It's very common for preclinical work to take several years to complete because of the extensive testing required by the regulatory agencies and by our internal review as well, because obviously we, even if they didn't require these things, we want to make sure that this is a safe and potentially effective drug before we take it to the clinic. So again, we note that we are still in the preclinical stage of research and development. It's possible, again, that our work may not successfully lead to a clinical trial. At present, the work is progressing as planned, and we're all very excited about that. But these are the steps, the painstaking steps that need to be completed in order to set us up for what will hopefully be a successful clinical program. So for UX068, animal testing refines our understanding of how the drug behaves so that we're equipped with this information in order to support a potential clinical trial as safely as possible. So a large part of the current work plan is to figure out how to dose UX068 as safely as possible without losing an opportunity to develop as much creatine as we can with each dose. So this means we have to understand how to measure creatine in animals so that we can interpret the creatine delivery on a human scale, and also to identify any potential negative effects that might be related to repeat dosing over longer periods of time. So these are just kind of, this is how the science gets done. It's painstaking, it's slow. We know it's frustrating for families, but we know that you understand that we want to have a safe and effective drug before we go to clinic, if we're able to go there. And so all of this work is used to eventually submit an IND, a new drug, investigational new drug application. And so all of these things, you aren't able to submit an IND without all of this painstaking work. So ultimately, if our preclinical work is successful and accepted by the agencies, especially the FDA here in the US, then we are hoping to be able to advance to conducting clinical trials. And we will work with regulatory agencies to incorporate their feedback along with the science that we've collected to determine the safest study design that allows us to collect the information we need to see if the drug is efficacious, if it's working. And the other important part of this is always feedback from the patient community and clinicians and researchers in this field to help develop our clinical plans. So we understand that you are eager to learn more, and we will share a timeline with you when it has advanced to the next stage. So I just want you to reassure you that our team is working with a sense of purpose and urgency to advance the research as quickly as we can, because we know how important this is. for your families, your children.
Thanks, Melanie, and we're certainly rooting for you and your team's success along the way.
Well, yes, and I'd say our partnership with ACD is absolutely vital in this process, and we really appreciate your cooperation and your all of the collaboration we've had with your team.
Thanks, Melanie. Last question on my list. Are there already eligibility criteria for participation in future clinical trials? It's got three parts. I'll say them all and then we'll break them down if need be. Second part, when is the next step involving patient participation in clinical trials? When are clinical trials planned?
Well, as I just said, we can't really tell you exactly when clinical trials are planned because we have to go through all of those steps that I just outlined. But we are currently in the preclinical phase, so we aren't yet there in terms of developing a clinical study protocol. And when we develop the protocol, that's when we will list eligibility criteria such as age, sex, whatever treatment the patient may or may not be in, other medical history that might exclude a patient from participating. For example, if they suffer from some other disease state that might affect our ability to measure things like, blood tests or ECGs or things like that. And then we will work on learning about CTD from patients and caregivers and expert healthcare providers as we continue to do. And we'll obviously get feedback from all the stakeholders I mentioned, including caregivers, physicians, researchers, and health authorities. So all of these things we anticipate and we welcome input from the community in terms of helping us design the best clinical trial. So that's why we come to the community and ask about things like, how does this population take oral medications, or how do you manage certain aspects of behavior or other aspects of the disorder. So these are things we come to you to ask frequently. And of course, the survey data that you're collecting will be really important. All of this goes into to making a successful clinical program. And we wouldn't at Ultragenyx, it's sort of our motto that we ask the patient community. We don't sit in our offices and decide on our own what needs to be done in a clinical trial. We go to the patients and the families living with this disorder to ask, what we need to consider when putting the trials together.
Thank you. Thanks so much, Melanie. So that was the end of the questions that had been pre-submitted. I see that we have a couple more in the Q&A box, and I think our plan is to be able to review them offline and provide written responses from your guys' team. So we've been keeping an eye out for those and we'll ensure that those are passed along to you. So at this time, I'd like to remind our attendees, take the last few minutes, please put in your questions in the Q&A box, and you can always also independently write to the ACD. If possible, let us know if there's other questions in case you're not able to submit it in the Q&A box, and we'll make sure that those are passed along to the Ultragenyx team. That is all I had on my list today. Any final thoughts, Melanie or Judy?
Well, again, we're so grateful for your partnership and so grateful to have wonderful investigators like Judy to work with. It's been a real joy working with her and the others on this study. So it makes our work so much easier.
I have really enjoyed working with Ultragenyx and with ACD. It's been, this is actually, it was hard to see that timeline and see how long we've actually been doing this. It doesn't feel that long, and it certainly feels like the urgency is there, and we're trying to do as much as possible in as short amount of time as possible. But the families have been really great to get to know and get to work with, and it just has really been very emotionally inspiring to try to understand this group and this disorder and the community around it. So you are always in our thoughts and in our work, and we hope that we'll be able to continue to see not only just regular development, but also it would be great if there was an enhancement with some intervention. It'd be great.
Amen to that. I think on behalf of the ACD and our community here today, I'd like to thank you guys for making time to be here at this event and address some of these questions. Really appreciate all of the efforts that you've taken so far and the commitment that you're showing to this today and in the future. Certainly on ACD's behalf, we stay committed. We're looking forward to whatever partnership opportunities there might arise, and stay true to the mission. So with that, I think we're ready to bid adieu and wish everyone a great weekend.
Thanks, everybody.
Thank you. Bye-bye.
Thanks all.